Today we're talking about this idea of precision health or precision medicine. So this is a popular term. A lot of hospitals, hospital foundations, there's a lot of hype around this idea of, precision health. When it actually comes down to defining that and conceptualizing what that means, I'm finding different groups, different researchers, different families, different clinicians have a slightly different take on what this term precision means. So I'm going to go over a little bit some ways of thinking about it from a neurodevelopmental lens. It's worth knowing the term was popularized actually in the Obama era around this idea that large scale clinical trials where you have 1,000 people in one group and 1,000 people in another group, and you compare them, will give you an average effect of a treatment, but it's not telling you for an individual within that sample, was this helpful or were there side effects, or it doesn't give you an individualized approach. So the idea of kind of precision health emerged as a way of thinking about what in epidemiology we call covariates, so sex and gender, race and ethnicity, other biological factors that might help identify which treatments are best for who. Now in development, it gets more complicated, right? So I'm going to walk you through some of the ways we're thinking a bit about this now, with a focus on the rare disorder space, which is where some of my research projects are focused. So a little bit of background. So for people who I haven't met before, so I see Alfredo, I see Andrada. Yeah, anyways, for people who don't know me, so I'm a child psychiatrist by training. I went to U of T for psychiatry and child psychiatry, and then did some focused training in development. And then I also did training in research. So my research is in a little bit in mental health and development, a little bit in genomic classification with respect to treatment decision making. So I'll talk about some of that today. So we're going to talk about the rise of the genomic era in neurodevelopmental conditions or neurodevelopmental disorders. And then I'll walk you through some of the things I find interesting about classifying and grouping, similar to what we're doing in epilepsy and cancer, for example. And then when it comes to this precision medicine, precision health question, it depends who you ask. I'll walk you through what some of the big data approaches are. And as a complement to that, what I'm interested in are kind of small N approaches. What can we do for an individual kid or a few kids? And then we'll summarize that at the end. This is my first time giving this talk also, so hopefully it goes smoothly. You are welcome to pipe in the chat in any capacity. I will try my best to watch it at the same time. So feel free to kind of jump in. Okay, so molecular era. So if we think of a group of, let's say, children and youth with neurodevelopmental conditions, in the 2000s, let's say you or your child was diagnosed with a developmental delay or intellectual disability, practice guidelines would say, you should do a karyotype. So look at all the chromosomes and see, are any, do you have Down syndrome or trisomy 21 or something like that? And then there were a few genes we could test for. So Rett syndrome, Fragile X syndrome, and tuberous sclerosis. And the rest of developmental conditions were conceptualized as idiopathic, which means we don't know the cause. And if you went before the 2000s, this is when you get into some of the honestly darker history of psychiatry that involves parent blaming and all sorts of kind of dynamic formulations of why kids might be developing differently or processing social information differently. In the 2010s, a clinical test called chromosomal microarray became available. And this is a blood test, so maybe some of you on the call or some of your families on the call would have gone through this experience where you get this new diagnosis of, let's say, autism. Your doctor says we can do genetic testing to find out if there's a variant associated with it. And this gave rise to kind of what are called copy number variants. These are chunks of DNA at these locations in the genome that are duplicated or deleted and are associated with different health conditions. Some of them with learning and development, some of them with heart or lung or other organ systems. But still, most of what we know about developmental conditions were kind of idiopathic, unknown. As of about the past 3 to 4 years, and what's kind of exciting in some ways is this individual, so genome-wide testing. So the microarray test, this one here, would be like if your genome was a library, we're checking to see our bookshelves duplicated or deleted or missing or doubled up. This one is going down, so genome sequencing and exome sequencing goes down to every single letter. And so this is, we're now seeing, and clinically in practice, we're seeing kids who are identifying with and identified by single gene disorders. So a variant in, let's say, ARID1B or CHD8 that explains a lot of their developmental differences. We still have a large proportion of kids where there isn't a clear genetic cause, or the genetic causes are multifactorial, or there's an interaction with the environment, but that proportion is getting smaller and smaller. And this is not just in MDDs, this is kind of broadly in health research. So if we're looking over, if you look at this graph here, phenotype would be like a condition. So a condition where we say, oh, sometimes babies are born and they have a heart defect and a cleft lip and cleft palate and small ears, and we know these things tend to go together. We don't know what causes it, but they tend to go together. And that over the years, we're starting to identify the precise underlying molecular cause. Similarly, now we have kids with different presentations. We're getting genetic testing, and we're being able to classify genetic contributors that didn't fit with a specific syndrome or picture. This is rising as well. And so if you look in some of these genetic databases, variants or genes that are associated with developmental impact, we have a list of about 1,000 or more presently, and that list is growing. And if you are to do one of these kind of fine resolution genome sequencing or exome sequencing tests to the letter, especially for kids with intellectual disability, the diagnostic yield, which means the chances of finding something that might help explain or understand health differences, is around 20 to 50%. So this number is not small. Now, there's lots of nuance to this, and I have to say, clinically in Ontario, we still can most of the time only get the microarray test. We're looking at that as a province, kind of what are the next steps for accessing the higher resolution, like the better test. And obviously not everybody may want or need this, but for some families, some individuals, especially when there are multiple health problems happening at the same time, it helps to make sense and understand what's going on for them. Now, this was a little project I did a couple years ago when I was still in training. And the take-home point is that genetic testing for any reason can help us understand development. So there is sometimes controversy around, like, should we be doing genetic testing for developmental differences? And my opinion on that is if that person wants it, then they should have the option to understand. But even if a kid's, let's say, undergoing a genetic test for a heart defect or for a kidney difference or kidney disease, what our study showed is of 3,000 kids at SickKids who underwent genetic testing for a non-developmental condition, 13% of them had a genetic finding, and of those 13%, half of them had evidence of a developmental impact or association with developmental disorder. And so the field is shifting so that increasingly very, very young babies and young families with let's say new parents, for example, are finding out all sorts of information about their kids' development and what might happen going forward from a really young age, often testing for another reason. Another complicating factor with the rare, what we're learning with this work is that there isn't a single, genetic gene or variant or anything like that that explains, let's say, intellectual disability or explains autism. it's complex, it's multifactorial. You've probably heard this before, and there are many, many, many different genes. And so this is an example from the complex care clinic at SickKids. These are kids who have high medical complexity, so often kids who are reliant on technology for either breathing or feeding, so G tubes, that kind of thing, and who have seen multiple medical specialists for many health conditions. And so in the complex care program, they have about 600 kids who have genetic diagnoses. And what you're seeing here is the most common group are kids who are the only one in the group who have that genetic diagnosis, right? So the most common genetic diagnoses affect, 5 or 6 kids, maybe 10 for 22q deletion. And so this creates a real problem for clinical trials and for research, because if most of your rare conditions are seen only once in your province, how do we make sense of that? How do we guide clinical care? And so it's a challenge for research methodology. So I'm a psychiatrist, right? So my interest is a little bit in when someone's presenting to me with mental health symptoms or behaviors, and they want to know about treatments, and they've tried other things, and they're wondering about medication. Can I use this genetic information in any way to guide what I'm doing? And so far, I have to say, not very much, but my hope is in 5 years, it'll be better, and in 10 years, it'll be better. And so one thing that is has interest is this idea that, maybe we don't need to collect every single rare condition and group them together and try every single clinical trial. Maybe there's a way we can group them in a way that's biologically meaningful, that might have treatment implications, even for kids who have, some genetic similarities. So you're starting to hear terms, and some of you may be connected to these communities, where genetic conditions are grouped by how their mechanism of action. So if you imagine over here, this is a picture of a neuron. So let's say that's your presynaptic and postsynaptic receptor, So think like grade 11 biology. In the boxes, the colored boxes are all genetic changes that can affect brain development. And we're starting to group them, for example, into scaffolding proteins. So these are ones that kind of anchor the two cells together. Things like SHANK3 you may have heard of, or GRIN2B. Or there's the channelopathy research groups who look at kids or individuals with variants in the ion channels, or those that regulate the epigenetic machinery, so which genes are turning on and off, or the ones that send the signals from the cell surface down to the nucleus. And so you're starting to see these clusters and research organizations kind of bringing people together. What's exciting about that for me is maybe there is clinical meaning to these groupings as well. So we've got all these kind of rare conditions clustered together, and maybe we might be able someday to say, oh, for kids with this subcluster, these types of treatments work better, or these types of treatments cause side effects, don't go there. This is an interest of some of the work I'm doing. And it's not just a pipe dream, because in epilepsy, this has started to happen, and in cancer as well, actually, but I'll go with epilepsy. Oh, I'm seeing chat. Sorry, I've been ignoring the chat. Lived through the 2000s, rare disorder identified in 2009. Okay, I'm going to save on those. I don't know TCF4, but thank you for your comment, Lynn. So in the epilepsy world, you're starting to see treatment decisions being impacted by genetic classification. So now let me move my head so I can see. So if you think about, so if you look at the gene column here, a lot of this is kind of, if you don't know these conditions, it's a lot of kind of Greek, but if you imagine there are groups of conditions where, let's say the mechanism leading to epilepsy has to do with your metabolism, and that certain vitamin treatments actually make a big difference in how often kids are having seizures. Or if you've got a gene affecting a channel, one of the signaling channels, potassium channels, sodium channels, then there are medications that act on those channels. And whether your your genetic difference either increases or decreases the functionality of those channels, it tells whether the medications will be helpful or harmful for your child. So much so that there's actually research trials looking at rapid genetic testing in neonatal epilepsy to try and make sense of, what are we doing right from birth to help these kids, as opposed to trial and error, which is how things are in mental health right now. There are challenges to this, though, and so hopefully many of them seem obvious from the description of the sample sizes here, but one challenge is heterogeneity is the term. So the idea that even in kids with the exact same genetic variant, their presentations are very different. Their clinical needs are very different. The degree that they're looking for medical intervention or not can be highly variable, and what their target symptom might be may not be the same. The one that often gets overlooked in these types of talks and studies is a term chronogeneity, which is kids look different over time. And I think in development, we know this more than in any other field, that you're when you're little, you might get a diagnosis of global developmental delay, and then ADHD, and then autism, and then depression, and then anxiety, and then something else. These are not biologically distinct bubbles of people. These are symptoms that change as we grow up. Hopefully the point about small samples is coming through from some of the data, that many of these conditions are super rare, and we're starting to develop networks for bringing together rare disease clinical trials, but across Canada, for example, but having the ability to both find people with these conditions and bring them together, but then also have methods to study them when you may only ever encounter 2 or 3 in the world. It takes kind of turning traditional trial design a little bit on its head. And then the other question is a little bit around, what are we measuring? And so just because, let's say, somebody's channels are firing better, does that actually equate a meaningful change in something that person wants changed in their life? And so we don't have good biomarkers for developmental outcomes. And our outcome measures, which often are surveys, and I'm sure by being in Pond, you've done a ton of surveys, sometimes they don't measure what we need them to measure. And so there is work being done, and important work being informed by families and participants about, are we measuring what's actually important? Okay, so I've done 20 minutes, I'm about halfway. So let's come down to some approaches to this kind of precision medicine question, and different ways people think about it. So if you ask a data scientist, what is precision medicine, they will say AI. That comes up a lot. Oh, it's AI. Artificial intelligence will mine all the data and tell us what works for who. If you ask a rare disease person, they'll say, it's gene therapy. Gene therapy is precision medicine. And so I'm holding these kind of different viewpoints a little bit in mind as I talk about this. The big data approach, Pond would be an example of a medium to big data approach. This is the idea, if we build large translational networks like Pond, like EU AIMS, and we connect and collect enough variables on enough people, then the data will speak for itself. And that's starting to be true in some situations where, we can cluster a handful of individuals who have similar biology, similar symptoms or behaviors of concern, and that maybe this can start to guide us towards an intervention for that specific cluster. You'll hear a term like massively multivariate. It's not like they picked, we're going to measure mood or we're going to measure language on everybody. They are going to measure everything on everybody. And there's starting to be some innovation with respect to clinical trial design, leveraging these big translational networks. And so Pond has been part of and Evdokia, who I'm sure you all know, kind of developing some of these. Outside of the field of development, this is happening and has been tried and often even led by in cancer. So it used to be you have a lung tumor, you go through the lung cancer treatment algorithm and you only trust data based on lung. As we've been able to understand the biology of cancer mutations and evolution, we're realizing that it's not where the tumor is necessarily, but how the tumor is made up. And so there are these big trials now. Example would be a platform trial where people get recruited irrespective of where their tumor is, but based on the molecular subtyping, and then they get randomized to treatments. And then if one treatment's not working, they cut it and they keep people moving through the pipeline, trying to get more and more precise. What you wind up with in the end are kind of very small groups of people with similar biology, but maybe very different cancer presentations. So in neurodevelopmental conditions, the idea would be maybe we're not looking just at, social differences in autism or focus in ADHD or anxiety symptoms, which affect everybody. Maybe we group everybody together and we try a treatment that might work, and we see, are there subgroups that it works well for? This is the CALM study, which is starting up shortly through the national network based out of Pond, where kids with different developmental kind of buckets or labels who are struggling with anxiety would all get the same treatment. And then we can check and see, how do they respond overall and how did the individual subgroups respond? So coming back to this AI piece, there's lots of hype in medicine around, can we, get the ChatGPT algorithms to look at all of the data and tell us the answer? And I wouldn't be the person to speak to, the biases or the challenges with that, but there's some interest, and we have a few projects starting up saying, if we can collect enough treatment information and enough biological information on individuals and how they respond, maybe without necessarily being able to have a hypothesis and a specific question, we can pull out some patterns. So this is an example of a project we're working on where we're taking health record data and Pond data, and we're looking to see if a kid comes to clinic with a mental health symptom or behavioral of concern, and they are looking to try medication for that child based on everything we know about them, which is the best class of medication to be tried, and seeing what AI might tell us compared to what the clinician might do. So we're nowhere near a state where, AI is going to tell you what medicine is going to work for your kid, but we're at the point that we're trying it out to see if maybe this would be a helpful tool. So I'm going to switch now to this idea of small n, or n of one, individualized trials, which is a growing interest of some of the work I'm doing, with the idea that maybe precision medicine has less to do with understanding everybody, but more to do with understanding one person and what can we know and inform our care for that one individual. So there's interest in the genetic therapy, so I'll talk a little bit about that. I will say I am, again, not a geneticist and I'm not a gene therapy doctor, so this is a bit of a summary as maybe a fan of some of that work or that research. But there are estimates that of rare genetic conditions being identified, like we talked about, rising rates, somewhere around 10 to 15% may be amenable to a treatment, a custom treatment targeting that underlying genetic difference. And big registries like Pond and others have been essential to building this work, because when a kid gets a genetic test done and there's a variant, much of making sense of that variant involves searching the whole world to see who else has that variant and what are they like. And so as people contribute to these large genetic databases, it helps contextualize and understand if these things are meaningful or not at all. And so you may see in the news talks about individualized therapies for specific kids. This family in Toronto, you've maybe heard speak for a SPG 50 is the gene, who has an individual genetic therapy developed related to developmental and motor delay and cerebral palsy. This child, Mila, had Batten's disease, like a degenerative brain disease, and they developed a precision therapy just for her, an ASO. So you may be hearing some of this discussion in the news, and these stories come around increasingly so. So you may hear the term precision therapeutics. I'm going to provide a quick summary of what that is based on this little cartoon diagram here. And I will cluster them into what we call ASOs, broadly gene therapy, these kind of custom molecules, which was on the previous slide. And then my interest would be in using medicines that we know exist and having them match to specific individuals or biological pathways. So if you hear this term antisense oligonucleotide, ASO, this would be from the previous slide for Melasin was the name of the medication they developed. The idea is if you have a genetic variant that you're born with, normally you would have that translated into messenger RNA. You'd have that transcribed at the ribosome, or transcription and then translation at the ribosome, and it goes into a protein. And that we can build a little complementary piece of messenger RNA called an antisense. So it's a matching piece of RNA, of mRNA, that would bind and catch that piece of genetic material before it hits the ribosome. So you can kind of trigger that for breakdown. There's a bunch of different mechanisms on how these work. But the idea would be if you can get the sequence for that condition and put these treatments into the cells, you can actually prevent or change the way our cells are making proteins. So this is ASO, and then there's something called small interfering RNAs, which also acts kind of on this post transcriptional pathway. Gene therapy is different. This would involve actually changing the genetic material in the nucleus. This could be through either trying to fix genetic change in right into the DNA, or inserting a little piece of genetic material that the cell's own machinery would then transcribe. And so both of these treatments have been developed for rare genetic disorders and are starting to be developed for individual kids with individual conditions. So on the previous slide, a gene therapy made for one kid, ASO made for one kid based on their biology. And you may hear this kind of precision medicine, and Sick Kids has a lot of fundraising around it. An example of how this is happening, if you see this little bubble down here, the N. Lorem Foundation, they are a non-for-profit that is making ASOs for kids with ultra rare conditions. And so there's a process where you can apply to have a custom molecule made, and you need to partner with an institution, but there's hype around that. So that would be kind of your custom or your bespoke therapies. So we talked about this. So the ASOs, the antisense, so this kind of like what it would involve, those ones are repeated treatments, often monthly. For brain conditions, often it's monthly spinal injection, so into the spinal fluid, to try and target the brain cells and affect that biology. And so we now have one that's approved for spinal muscular atrophy, and they're being investigated for some neurologic conditions like Angelman syndrome, for example, which is a neurodevelopmental condition. Gene therapy is kind of exploding, and my understanding from a workshop I was at today, what you read in the literature is already out of date, like how fast this is turning over. Mostly it's what's called ex vivo, so taking the cells out of the body, mucking around with them with gene therapy and then putting them back in. So for cancers or immunologic conditions, we do now have an approved gene therapy treatment for spinal muscular atrophy. Some countries, Canada not yet, for AADC deficiency, which is a metabolic but a brain and neurologic condition. And they're being investigated for a number of other kind of brain-based conditions. So I think in the next 5 years, there's capacity building happening, and also the basic science is really moving at quite a fast pace there. The challenge is, even if we developed, the perfect molecule for whoever, for whatever genetic condition, we still need infrastructure, we still need medical expertise, we still need management of all of the procedures and side effects that go with that. And then we need robust trial design to determine, is it actually improving what we want it to improve or treating what we want it to treat? It may not matter if the number of your cells expressing a protein increases if you're not seeing, benefits for whatever your target symptom might be. So there is a body of science around clinical trial design for small N, so small samples, or N of 1 studies. If you look here, a classic N of 1 design is one where you would start someone on a treatment and then you'd switch them, treatment, not treatment, treatment, not treatment, and you do it in a blinded fashion. And the more times you do this, the more confident you are that you're having the effect you wanted to have, or the treat the symptom you wanted to treat. The challenge with some of these kind of bespoke molecules or gene therapy is you can't take it away. Like, this is, you're committed to this. So we need to be a little bit inventive about which some people would call kind of like quasi-experimental approaches to make sure we're rigorously tracking how people are doing and measuring what's important for families to measure. These are some projects that I have started using different approaches. So in kids with very rare conditions, studying existing treatments, so existing agents that are available on the market that we think might have a different impact on them because of their unique biology. So this, for example, is a condition where your brain doesn't make neurotransmitters. What would happen if someone took an SSRI, for example? This is a condition where the channels are overfiring, and what would happen if we gave a medication where we knew it slows down that overfiring? And so designing little studies for individual kids to see, can we make meaning out of their treatment journeys as we're going through. This condition it affects how genes turn on and off, and we can actually measure that turning on and off of genes. So we wondered whether in ARID1B, as someone's going through their treatment for mental health, does it leave a signal that we can measure? So in summary, so far, there have been amazing advances in genomics, technology, data science, precision therapies. Large cohorts and registries like POND have been essential to this progress, both to understand the prevalence of these types of genetic changes and to contextualize and make meaning of different findings as we're moving along. So far, most rare genetic diagnoses are not directly impacting everyday treatment decision-making in neurodevelopmental disorders. There's some exceptions to that. So there's some conditions where kind of less rare of the rare, where we are starting to understand natural history and what treatments might respond differently. But for the most part, and I hear this from families all the time, like, wow, great, I have this new condition label. What do we do differently? And the answer often is we don't know yet. Although I have to say the rare disease family communities have really pushed the needle in that way. I would caution, although there's lots of hype around the custom genetic therapies, they're still very experimental. There's still a handful, of kids in Canada that this is realistically happening for. The spinal muscular atrophy story is an example of a gene therapy that drastically changes kids' developmental course. And in Ontario, through the newborn screening program, I think they've treated 35 kids, something in that range so far. So it's not a mainstream thing at all yet. And we do need capacity, expertise, and really infrastructure to do this properly. We can have all of the investment in the cells and the animals and the biology, but if we're not actually moving the mark for the families taking the treatments, and we're not sure what we're measuring, it kind of brings the whole thing in question. So I think this is where I'm hoping some of the work I'm doing can be helpful in the longer term.